So we capture a bat, we inject it with the vaccine, and it can then fly back to its colony, and it will infect those bats around it with the vaccine.
Welcome to the Knowledge Archives podcast. We're a group of students on a mission to learn from as many different disciplines of knowledge as possible. I'm your host, Madhav Malhotra, and today I'm glad to be joined by Megan Griffiths, a PhD student at the University of Glasgow in Scotland. She researches an innovative tool for public health, self-spreading vaccines. Today, she explains the history of using different vaccination techniques in wildlife and how self-spreading vaccines could innovate atop all of these. Before we dive into, all of the technical details, of which there are many, I'd love to just ask you in general, what is your background? What led you to this area of research?
Yes, so my name is Megan, and I'm about to finish my PhD at the University of Glasgow Centre for Virus Research. And I did my undergraduate in Cambridge, and I started off doing a course called Natural Sciences, which is very broad. I started with chemistry and cell biology and also physiology, and I sort of became more specialized throughout the course. And I eventually settled on virology and epidemiology as my two favorite things. And also during this final year course, I came across what is my favorite virus, which is rabies virus. Most people don't have a favorite virus, but I think if you speak to more virologists, you'll find that they all have one. And then when I was looking for a PhD, I came across Daniel Stryker's lab, which is the lab group I'm currently in. And the lab group works on looking at rabies virus in vampire bats. And it's just such a good combination of both virology and sort of animal behavior that it really caught my attention. So I sort of decided to lean into that project. And at the time that I started, a previous project had just finished, which was looking at a slightly different type of vaccine, which hadn't really been explored in a sort of practical subject before.
Mm-hmm. So it seems like trying lots of undergrad courses, it really benefited you, and that's nice to hear. To talk about your research now with vaccines, most people aren't even aware that we have vaccines that aren't used for humans. Could you talk about why we even vaccinate animals and what that concept is about?
Yeah, so animals have actually been vaccinated for a really long time. Starting from a human point of view, people can think of their pets. If you've got dogs or cats, you will take them to the vets and they will become vaccinated. This is kind of for two reasons. So first of all, to protect the health of the animal itself, especially in your pets. There's lots of diseases that can kill young animals in particular if they get them when they're puppies or kittens. And also they get vaccines to prevent them from passing diseases to people. A good example of this is the rabies vaccine, which is given to all dogs because rabies is incredibly deadly. And livestock, another sort of animal which is very close to humans, are also commonly vaccinated. In fact, a livestock disease called rinderpest is one of the biggest success stories of vaccination next to smallpox, because it's the only other virus which we've managed to completely eradicate by vaccination.
So if I understand that correctly, the rabies virus, if it infects a dog and then the dog bites a human, then the human can have detrimental health. So we're figuring out that indirectly, if we promote the health of the dogs, then there's less opportunity for these diseases to be transmitted to humans.
Yes, exactly. Yeah, that's a really good way of putting it. Obviously, we focus on dogs because humans have a lot of contact with dogs as pets, but humans also contact other animals in the environment. And also importantly, wild animals contact our pets, who then contact us. So there's this whole transmission chain of diseases traveling from wild animals into humans, either directly or via our pets.
Yes. And, people might have seen all the headlines about COVID-19 in bats. So could you tell us a little bit about Why is this risk from wild animals increasing for human populations?
So this is for a couple of reasons. One of the biggest ones is that the human population is always expanding. 100 years ago, we had far fewer contacts with wild animals because there were fewer people, especially encroaching into the sort of subtropics region where there are sort of a very high diversity of wild animal species and therefore a high diversity of viruses that they happen to carry. So the human population is expanding, and this comes with things like deforestation. And this is for often like livestock farming. So this really changes the natural environment and puts the wild animals that used to live there under a lot of pressure. So they might move more into human areas for scavenging, for example. And this just really increases the number of contacts that we're seeing.
Yeah. Could you tell us about any historical tragedies or, success stories of vaccinating animals?
Yep, yep. I think actually I'll stay with my area of expertise, which is rabies, because rabies vaccination in Europe and North America has actually been hugely successful. So in Europe, it's mainly the red fox which carries rabies as a wildlife reservoir. And a wildlife reservoir means that this species can maintain the virus just within the species. Even though it can transmit it to other species, it doesn't need that for the virus to sort of stay around. So I think it was in the early 70s, a lot of wild dog vaccination and pet vaccination had been used to try and reduce rabies cases in Europe. But the experts realized that rabies was still emerging into livestock and humans. So they were like, where is this coming from? And eventually they narrowed this down to red foxes and decided that they really needed to be vaccinated. But it's very difficult to vaccinate wild animals because they live in the middle of nowhere. You can't round them up very easily. And there are quite a lot of them, so you don't want to have to inject them all individually. So they made a vaccine formulation which was contained in a food bait. Initially, this was basically vaccine stuffed inside chicken heads, which were then dropped from helicopters into the wilds of Europe and left for the foxes to feed on. And this was really effective. I mean, it did take many years and sort of lots of follow-ups to work out where they were failing and succeeding in this vaccination program. But for a lot of Europe, rabies is no longer a problem. So it was very successful.
Would you be able to give an overview of like some of these traditional methods that we've been mostly reliant on over, let's say, the past 50 years in terms of how do we go and vaccinate these animals?
Yes, yeah. So as with humans, using injection is sort of like the first way of thinking about things. This is what's going to be used in your clinical trials to make sure that the vaccine actually works. So if you have like a test animal in a lab, you will use injections because this is the best way of sort of working out what dose of vaccine you're giving to an animal. For I think nearly all cases for animal vaccines, they then do move on to making them orally available, which means that the animal can eat the vaccine and then it's absorbed via its gut. For some vaccines, this isn't possible, so they have to remain with injectables. Then this is because the stomach is highly acidic, and they have to have sort of proper protective coatings to prevent it from being broken down before it can be properly absorbed and create an immune response. But for things like rabies, the researchers need to work out what foods are going to be most appealing to the animals. This is quite challenging, especially if you're targeting multiple species. In North America, lots of different species carry rabies and they all like different foods. So they've tested things like little cubes of sort of ground up fish meal with vaccine inside. They have like pellets of different sizes for different size animals. So there's a lot of research that goes into finding out what kind of food is going to be most appealing, but will also deliver vaccine appropriately. And then there's the issue of getting it into the environment in ways that the animals will actually approach. You don't want anything to be dropped too dramatically because it's going to sort of scare animals away from an area. Also, there's the big issue of not having just one single animal eat all of your vaccine and not leave any for the rest of the animals in an area.
Dropping food chunks everywhere in a forest, it seems like it's very complicated, as you're highlighting. Why why bother? What are the benefits that people see with that?
So it has been quite effective. And even though there's sort of lots of steps to go through and working out the best formulation, it's still much lower effort than catching all of your animals individually and injecting them, as well as sort of being cheaper and needing fewer people involved. It also means there isn't this sort of direct contact between animals and humans, because if you're physically catching animals to vaccinate them, then there's a lot of risk of disease transmission during that physical interaction. And that's not just from animals to humans, but also in the other direction, from humans to animals. You may have heard during the COVID-19 pandemic that there's been a lot of worry about zoo animals, such as tigers, these endangered animals, catching COVID and dying because they're endangered, so this is a big problem. So we also want to protect animals from our own diseases.
Well, it seems reasonable. Shooting food in a forest, it has, it's better than individually injecting animals, but when it comes to your research on self-spreading vaccines, maybe that might even be better. Could you tell us about kind of the motivations and the basics of what self-spreading vaccines are?
Yes, so the reason that we're considering self-spreading vaccines for my system, which is vampire bats, is because we can't use food baits on them. So vampire bats drink blood. It's their only source of nutrition. They just drink blood to survive. And this means that you can't really leave food baits out. Bags of blood dropped in the Peruvian mountains aren't particularly effective because it doesn't exactly stay fresh for very long. So we need a different way of getting vaccine to these bats. So self-spreading vaccines are basically formed of two main components. You need a bit of the pathogen we're concerned about, such as rabies in this case. We take its glycoprotein. So this is a protein that is found around the outside of the virus, and it's the main bit that our immune systems recognize. So we take the DNA that encodes this protein, we call it an antigen, and we need to put that inside a different virus. And this second virus needs to be harmless because we don't want it to kill our bats in any way. And it also needs to be very host specific because we don't want this vaccine spreading outside of the target population. So we've got this harmless host specific virus that can circulate in vampire bats. And if we genetically modify it to carry the antigen from rabies, it can now act as a vaccine. So when you put it inside a bat, this virus will replicate and during its normal life cycle. And whilst it's doing this, it will produce the rabies glycoprotein, and the immune system will see this and create an immune response, meaning that the bat is immune to rabies. Because this virus can replicate, it can also transmit to other bats. So we capture a bat, we inject it with the vaccine, and it can then fly back to its colony, and it will infect those bats around it with the vaccine.
So from like a general understanding point of view, what I might say is we have two key ingredients. One key ingredient is that we need some kind of virus that can spread in just this animal that we want to target, and it shouldn't harm that animal. And then the other key ingredient is we want to find some instruction within another harmful virus that will tell us about what I'll call flags that this virus wears so that the immune system can recognize, hey, this is like a red flag virus. We wanna get rid of this. And so we take those instructions in the red flag virus, we put it in the healthy virus, and then these instructions tell the healthy virus to also make red flags that the immune system can recognize.
Yes, that's correct.
So first thing is like how can a virus be healthy? We're all told that viruses are bad. How can an animal just be infected by some virus and not be harmed by it?
Well, so we're full of bacteria all of the time, and we're also full of viruses all of the time. And most of the time, they don't do anything to us. The thing is that this isn't very widely studied because they're harmless to us, so we don't really care about them. And other species have the same thing, and they might be different to the ones that we have. So sometimes a virus that does nothing in one animal will be harmful in something else just because they're not used to it. Kind of like if you go abroad and drink the water there, you might get sick because of the bacteria in the water because you're not used to being exposed to it.
Would you want to comment on the remarkable immune system of the bats and how they can be virus infected all the time?
Yeah, so this is actually kind of controversial within the bat research field. So it is definitely true that bats are able to survive with lots of viruses that are really harmful to humans. And there's a lot of research going into how their immune systems might be different to humans and other species. But they are definitely harmed by some viruses. And some new research is showing that we just sort of maybe aren't looking in the right places, because as humans, we're mainly interested in the diseases that we share rather than the ones that might just be harming the bats.
That seems like a interesting area of research that will keep on resulting in more novel findings.
Mm-hmm.
To get back to this idea of self-spreading vaccines, though, could you tell me about the pros and cons of these vaccines in this particular context?
Yeah, of course. So, first of all, everything that I've done is very preliminary. So, we don't know if it's going to work yet. But let's say it does work, and that the virus we've chosen to be this vector to carry the vaccine, it should spread well between bats. It should only infect bats. So, it shouldn't spread to humans or livestock or other close contacts that they have. And this means that the vaccine should be able to move well through the bat population and vaccinate a really high proportion. My results show about 80% vaccination could be achieved using this vaccine. And that's just using sort of like a one-time effort where you capture a group of bats, vaccinate them and release them, and then it just does the rest by itself. So this is a massive advantage because it really cuts down on the costs involved and the number of people, and importantly, having to go out into the wilderness, into caves, through rivers, through very spiky forests, trying to find the bats. So that's one big advantage. One of the main disadvantages is that there is a lot of concern over, first of all, should any vaccine or viruses be manipulated? And especially if you are manipulating them, should you then send them back out into the world? And that's a very understandable concern that people have. I would say for this system, you don't need to be massively concerned, because the virus that we've chosen is very host specific. It's a herpes virus, and these actually infect lots of different animals, but each one is specific to the species that it's sort of evolved with over time. So humans have herpes viruses, and mice have herpes viruses, monkeys have herpes viruses, but you can't infect a different species with your own herpes virus. So this shouldn't be able to get into any other species at all. It shouldn't be able to infect people or even other bats. And as for the genetic modification we're doing, the only thing we're doing is adding in this red flag, as you called it, into the DNA of the virus. And it doesn't belong there. So over time, what this herpes virus is likely to do is actually kick out that red flag. It's going to remove it and just become its normal self again. So that's the sort of evolution we're expecting to happen. Some people might have heard of the polio vaccine, and there have been a lot of problems with the polio vaccine in the past because it was a live attenuated vaccine. And this means that the pathogenic polio itself, which causes a range of symptoms from like mild colds all the way to paralysis and death. So they took this live virus and kind of turned off some of the more deadly bits of it. And then they used this as a vaccine. However, this was done quite a while ago when perhaps genetic techniques weren't quite as good as they are now. And they didn't quite realize that this vaccine would evolve over time and actually turn those deadly bits back on again. So this created a version of poliovirus, which is called vaccine-derived poliovirus, and it's now circulating separately. So that was a sort of a bit of a backfire in vaccination. However, because we're starting with a harmless virus, there's no deadly bits to turn on. So the worst that evolution can do to our virus is turn it not into a vaccine anymore.
I understand we're getting into more skeptical territory, and different biologists will have different points of view. That said, I will just ask you for your personal view.
Mhm.
In science, we have this concept of dual-use research, where we want to not only think about these application-specific pros and cons, but also if this paper gets published, if this knowledge is out there in the world, what are the larger drawbacks and potential benefits that we can get as an entire society? Some people are very concerned about this kind of research, when we figure out how to use these technologies, then people might get afraid. What if this happens in humans? What if we're vaccinating humans? One human is vaccinated and that spreads without others' consent. So could you just, from your own perspective, of course, tell us about your thoughts on the dual use benefits and drawbacks of pursuing this research and why you decided that it's still worth pursuing?
So, first of all, I think I can say that a vaccine like this would never be approved in humans. It's always going to remain an animal vaccine for the sort of bodily autonomy, consent reasons you mentioned, and also because in human health, we care a lot about the individual health, whereas in animals, we're sort of more concerned about the population immunity. So even if you found a suitable harmless virus in humans, there are a lot of immunocompromised people in the world, and we wouldn't want this vaccine anywhere near them. So it's not going to be, I think, a human issue. As for the dual use, I think a lot of the technology we're using actually already exists. We're kind of just applying it in a new way. So technology to genetically modify viruses has been around for like decades, and it's already used to make non-transmissible vaccines. So I guess the next step we're taking is just just putting that red flag in, as opposed to where current vaccines will put that red flag in and also make the vaccine not able to replicate properly. I think other places this could be used in, I think expanding it to other disease systems will be really useful. There are a lot of places where there aren't current vaccines for really deadly diseases, especially ones that come from wildlife. Things like Ebola. We actually don't know what the proper reservoir of Ebola is in the wild. There are theories of it being certain bat species. And if that's confirmed, that would be a really good thing to tackle, not just for human health, but for animal conservation. Because when animals carry diseases, this makes people very scared of them and more likely to kill them, which is obviously not great for the animals. Also, there are lots of endangered animals which carry their own diseases completely unrelated to us. And it would be really great if we had a way of helping them without sort of putting ourselves at risk with contact with very large, dangerous animals.
I appreciate you providing your perspective on this tricky question. I understand that other people, obviously, they might have different perspectives. So I appreciate you, taking the risk to, walk us through your current insight on this. And also, I understand that, this is an immensely complicated area. So I want to thank you for trying to keep this simple and, talking to us about this hugely influential discovery that could create a lot of pros and cons that we need to think about for various organisms, including us humans in the future. So I very much appreciate you making the time today.
You're welcome. Thank you for having me.